my name is Majid Ayaz. I am from Pakistan and I completed my master degree in Taiwan in Asia University at the Department of Medical Laboratory Science and Biotechnology under the supervision of San Chi Chen. And my research topic is epidemiological analysis of major cucurbit infectious orthotospovirus in Taiwan. So, and in this PowerPoint, my study is on with background and taxonomy, study objective, and association with the virus that I selected for in my study, and also result in discussion. So, background. So, in the production of cucurbit production of production of cucurbit in global and Taiwan is have a lot of importance because in throughout the world 222.3 million tons is the production of plants, and more than 75% as production is concerned in Asia. And there is three major crops I study in my this research is watermelon, cucumber, and melon. And in Taiwan is 221 tons, which the value is almost 6 billions. In the output value is 6 billions. So next is why cucurbit are important in Taiwan. Because it's a lot of farmers culture the different cucurbit plants, and a lot of people likes to eat the watermelons and melons in Taiwan. And major areas like Hualien, Tainan, Yuelin, Pingtung, and these areas are the most important agriculture area in Taiwan. And these are infected with many viruses. More than 90 virus has been reported to the naturally infected cucurbit virus, making regular virus monitoring and early molecular detection important for the protection of these crops. So next is Taiwan production. In the different area is the different production. Like in Hualien, the watermelon production is 35 point, is around 3 million kg is the production of watermelon. And like in Pingtung and the Changhua is a cucumber, and in Changhua city also melon production is also on the peak. So symptoms of the cucurbit viral disease is like in melon they show the yellow mosaic interval coloraces, molting leaf coloring, surface roughness, and yellow green um, discoloration. And in watermelon, it showed the severe mosaic coloraces and leaf coloring, deformation, molting, and reduced leaf expression. And in cucumber, it showed the coloraces, intervennal coloraces, yellow and green mottle mosaic and pattern, and reduced leaf size and leaf deformation. And what is this? My virus is MYSV and WSMoV. MYSV is melon yellow spot virus, and WSMoV is watermelon silver mosaic virus. These are both are orthotospovirus viruses in Taiwan. They mostly infect the cucurbit plants, and their symptoms is mostly common like chlorosis, mosaic, mottle, leaf color deformation, necrotic spots, and reduced plant growth. So visually we can't identify the crops. So that's why the molecular detection is very important to find out the detection of the which virus infected by the plants. So uh, and the and the most important is they can be transmitted by that vector which called is Thrips palmi in a persistent manner. So taxonomy and biology of Orthotospovirus, basically the taxonomy of MYSV and WSMoV, these are belong to the genus Orthotospovirus, and the family is Tospoviridae, and they are mostly 90% of similarity among these two viruses. So previously, before 2016, they are classified in genus Tospovirus, and in a family is Bemoviridae. And after in 2016 is updated to rename as a family is Tospoviridae, and the genus is Orthotospovirus. And so far, 30 species is recognized, and they are three serological groups: WSMOV group, TSWV group, and IYSV groups. They have different viruses and the taxonomy and also uh, they have similar disease, similar symptoms of infections. So biology of orthotospovirus, basically they are query shape envelope virons about 80 to 120 nanometer, and they have a single stranded RNA, is lRNA, mRNA, and sRNA. lRNA is viral replication and transmission, help in viral replication and transmission, and mRNA is cell to cell movement and also Thrips transmission, and sRNA is suppress the host defense and form nucleocapsid. And these trips acquire viral mainly as a larva and transmit as adult. And they have main key protein is viral replication and transcription protein, and like the silencing suppressor and glycoprotein and movement protein and nucleocapsid protein. So these are the mostly known traits that are transmitted orthotospoviruses. So propagation of transmission of trips, there are four stages: acquisition, replication, adult stages, and new infection. Acquisition is larva trips feeding on infectious plant tissue, and then they replicate virus in persistent manner and replicate inside the vectors. And adult virophilus adult disposed to new, and then it's infect to another new plants. Virus is transmitted during the feeding, and WSMoV and MYSV host in Taiwan. Their hosts are mostly similar, like watermelon, melon, muskin, wax ground, cucumber, and pumpkin. Like these are the host of um, Orthotospovirus, like MYSV and WSMoV in Taiwan. So these are the important cucurbit virus in Taiwan. They are known so far, like zucchini yellow mosaic virus, watermelon silver mottle virus, cucurbit coloratic yellow virus, melon yellow spot virus, and squash leaf Philippine virus, and papaya ring spot virus, and cucumber mosaic virus. And the new virus is cucurbit coloratic virus. So significance of MYSV and WSMOV in agriculture. They did this virus infect the yield and quality loss, and is diagnosis challenge in the field. You can't diagnose on the naked eye. You have to do the molecular detection and also vector divert spreading. So study design, objective of my study is detection of this virus using RT-qPCR, compare virus distribution by year, months, and crops and region, and evaluate the virus and also mix infection and generate the updated epidemiological information to support surveillance and disease management in Taiwan. So these are the sample collection of my sample collection in 2 years. And the diagnostic workflow is field sampling, RNA extraction, RT-qPCR detection, and positive and then analysis. So result, these are the symptoms that I observe in the field, and then I further do the purification and then detection of the virus. In the annual detection rate, the 357 sample of 2024 I collect, and in that 99 sample were MYSV positive and 78 was WSMV positive, and in 65 samples is the mixed infection. ASM, like in 2025, 243 samples, in that 38 are MYSV and 27 as WSMV positive, and in these 10 our mixed infection. So this the virus is associated with three cucurbit crops like melon, watermelon, and cucumber. And the total sample of sample size was 87 of melon, and 439 was watermelon, and 74 was cucumber. In this, the higher MOSV rate in the cucumber as compared and also the higher MOSV rate and higher proportional mixed infection in the cucumber. In the in the watermelon, the both viruses are similar overlap the level, and in melon MYSV is higher and WSMOV is increased in 2025. Like I will show you in a further separate yearly distribution of the virus. So the virus associated with melon in 2024 the sample size was 42, and in 2025 the sample size was 45. And this MYSV was 29, and WSMV is in 4 sample was positive, and therefore the mixed infection was also 4. In 2025 the 2 positive with MYSV and 70 was positive with WSMOV, and the mixed infection was 24. And WSMOV become more common in melon during 2025, especially in the April month, because it's the most architecture month in the Taiwan. The farmer grow the melon a lot as compared to the other months. In association with the watermelon, the watermelon production in Taiwan is very high. So so is the main reason that my sample size of the watermelon is also high. So and these are in 2024 is 272 samples of my size, and the WSMOV was and 650 samples, and 63 sample, and MYSV was 50 sample, and the mixed infection was also in 50 sample. And into 2025 167 sample was collected, and that 24 was MOSV positive and 9 was WSMOV positive, and the mixed infection rate was 7. So the overall the MOSV and WSMOV detection were nearly similar in watermelon. Mixed infection were numerically high in watermelon because watermelon has the large sample size among my study. And association with the cucumber in this 43 samples was my sample size in 2024, and 31 in 2025. In that 20 sample was positive with MYSV and 11 was positive with WSMOV, and the 11 are also the mixed infection of WSMOV and MYSV. So in 2025, the sample size was 31, and 12 were positive with WSMOV, and 1 was positive, 12 were positive with MYSV, and 1 was positive with WSMOV, and the mixed infection was also 1. So WSMOV was the dominant virus in cucumber in both year, and the WSMOV was more lower. And the cucumber had the higher proportion mixed infection rate among these 3 crops. So these are the seasonal pattern and monthly incidence. Virus incidence was generally higher in 2024 than in 2025, and the watermelon showed the strong infectious pressure in June and August in 2024, and mixed infection appear mainly during the month with high virus incidence, and seasonal peaks likely relate to the host availability, crop staging, warm climate, and the Thrips vector activity. So the mixed infection of WSMV and the MYSV was like in the whole 600 sample size, but the mixed infection was 12.5%, like 75 sample was positive with mixed infection with WSMV and MYSV. So mixed infection can make field symptoms more variable. Molecular detection is needed to become co-infectious sample may not be identified by symptoms alone. So discussion and conclusion. So the both in the discussion the both virus was detected in Taiwan during 2024 and 2025, and the MYSU are more highly associated with cucumber and melon, while watermelon support both virus at similar level. Year to year variation suggests the virus pressure change with crop composition, sampling a month, environmental condition, and vector activity. Mixed infection was present in all 3 crops, supporting the need for different molecular diagnosis, and RT-qPCR with melting curve validation strengthened detection reliability. So disease management, so we can monitor the virus earlier stages and reduce the sources, use healthy seedings and remove infected plants, and manage the vector, burn and monitor the trips, and rotate insecticides to slowly resist development, and confirm the testing with the qPCR. In the conclusion, the study confirmed the occurrence of WSMoV and MYSV in melon, watermelon, and cucumber in Taiwan, and MYSV are more common in cucumber and melon, while watermelon showed the most nearly similar detection level for both viruses. Mixed infection occur in all these three crops and was proportionally high in cucumber, and virus incidence were varied by crop, month, year, and um, highly pressure in some months of 2024, and these findings support seasonal specific surveillance and integrate management of cucumber orthotospoviruses in Taiwan. So these are the reference, and thanks to my professor and my lab mates and my friends in Taiwan for motivate me to do the study and help me to complete my master degree in Taiwan. Thank you all.